pituitary gland. On today class, we'll study uh, hormones which is secreted by posterior pituitary gland. So on today's session, we'll discuss about the posterior pituitary hormones. So in anterior pituitary gland, there are 6 types of hormone are synthesized. In posterior pituitary gland, there are 2 types of hormone synthesized. So the major these 2 hormones which is synthesized in posterior pituitary gland are this vasopressin and oxytocin. So vasopressin, so that is the hormone which have a antidiuretic actions and the pressure actions. So its main function is the regulations of water in the body. So another hormone which is synthesized by posterior pituitary gland is oxytocin. So which is involved in a contractions of smooth muscles of mammary gland and uterus. And the common things of these 2 hormones is both are small peptides which contains 9 amino acids. So that's why they are also called as a nonapeptide. So nonapeptide means that contains 9 amino acids. And they both are synthesized in the neurosecretory neurons. The very little difference among these vasopressin and the oxytocin. So the amino acid sequence of this vasopressin and oxytocin is quite similar. Only differentiation is that is the 2 amino acids that is present on 3rd and the 8th position. So for the both hormones, that is the vasopressin and oxytocin, the common ancestor is that is the vasotocin. So though they this unlike anterior pituitary gland, the posterior pituitary gland, it do not contains the cell, and generally this the posterior part of the pituitary gland is the axon of a cell, hypothalamic cell. So that is the magnocellular cell. So that's why the hormones which is secreted by posterior pituitary gland, so actually they are synthesized in the hypothalamus. So the cell body part of the cell lies in a hypothalamus. So this vasopressin and oxytocin, so both are synthesized in hypothalamus, and the cell's axon is end on the posterior pituitary gland. So that's why these hormones are secreted and released from a posterior gland. So these hormones, this vasopressin and oxytocin, both are synthesized in the cell bodies of magnocellular neurons of supraoptic and paraventricular nuclei of hypothalamus. So this oxytocin and vasopressin, they both can be synthesized in the both nuclei, that is in the supraoptic and paraventricular nuclei, though their cell is different. However, this supraoptic nucleus, they predominantly synthesized antidiuretic hormone or vasopressin, and paraventricular nucleus synthesize oxytocin. So these both cells are able, these both nuclei are able to synthesize both hormones. So that is the supraoptic nuclei, it can synthesize both this vasopressin and oxytocin, like this paraventricular nuclei also can able to synthesize both hormones, that is the antidiuretic hormone, that vasopressin and oxytocin. However, this supraoptic nucleus, it predominantly synthesizes vasopressin, and paraventricular nucleus is predominantly synthesize oxytocin. So like other these protein hormones, these hormones, this vasopressin and oxytocins, both are synthesized as a hormone in a nucleus. So in a cell, they are synthesized like other proteins. They are also synthesized by a ribosome, and during a synthesis they are synthesized as a pre-pro hormone. And these pre-pro hormones, when they enter to the this endoplasmic reticulum, so it loses the signal peptide and they convert it to the prohormone. So in the form of the prohormone, they are packed into the these vesicles. So on vesicles, with the help of the proteolytic enzymes, these prohormones, they are cleaved into the active hormones like this one, vasopressin and oxytocin, and they are secreted in the form of the active hormones, that is in the form of this vasopressin and oxytocin. So you have studied during this one protein synthesis, or this during a protein targeting. So generally these protein are synthesized in a ribosomes, and after the synthesis in the ribosomes, they enter to the endoplasmic reticulum, and from endoplasmic reticulum they are packed into the vesicle, so that means in the Golgi apparatus, and from that vesicles they are targeted to the targeted site, so they are transported to the targeted site. So simply like that, in here also these hormones, they are during a synthesis, they are synthesized as a pre-pro hormones, and during this pre-pro hormone they contains a signal peptide. With the help of this signal peptide, these hormones or these proteins, they enters to the endoplasmic reticulum, and as they enter the endoplasmic reticulum, it removes a signal peptide sequence from an N terminal, and so that these pre-pro hormone, the pre-pro proteins, they are converted to the pro proteins or pro hormones, and in the form of pro hormones, they are packed into the vesicles, and from endoplasmic reticulum, in the form of vesicles, they are enters to the Golgi apparatus, and in the vesicle itself, with the help of the proteolytic enzymes, they are cleaved. These pro-hormones are cleaved, and they are divided into the active hormones and the other peptides. And when there is a necessity, and they are released from vesicles. So from the vesicles, they are transported on the axons of this these nucleofibers, and they are stored in the this one posterior pituitary gland. So during the formations of these pro hormones, this ADH is associated with a neurophysin 2, and oxytocin is associated with a neurophysin 1. So now we'll move on to the individual hormones. So this oxytocin. So it is a cyclic nonapeptide which have molecular mass of approximately 1000 Dalton, and it form a cycle with the help of a two amino acid, that is a cysteine. So two cysteine molecules. So first cysteine molecule, it form a disulfide bond with the 6 positions cysteine molecule, so that it form a cycle form, and the remaining 3 amino acids are joined. So the difference on this oxytocin and vasopressin is these third positions, that in oxytocin, this ileum, the isoleucine is present in the third positions and leucine is present in the eighth positions. And in vasopressin, this isoleucine is replaced by this aspargine and this leucine is replaced by arginine. So these hormones, when they release from a posterior pituitary gland into the circulations, they moves in the free form and they bind with the specific receptors that is present on the target cells. And these hormones are rapidly degraded, mainly in liver and kidney. So the half-life of this oxytocin is around 3 to 5 minutes. So apart from this posterior pituitary gland, this oxytocin is also synthesized in uterus, placenta, amnion, corpus luteum, testes, and heart. So apart from this posterior pituitary gland, this oxytocin can also synthesize in the other parts of the body, like the uterus, placenta, amnion, corpus luteum, testes, and heart. So the mechanism of action of this oxytocin is G protein mediated. So in the target cell, the specific receptors are present in the target cell. So the specific receptor for oxytocin is present in a target cell, and generally these receptors are these heptameric receptors. That means which contains a 7 protein complex, and generally these receptors, they are G proteins. So on G protein, they are made up of, that is the alpha, beta, and gamma. So what you have studied during the how this G protein works. So here this specific receptor for oxytocins, which is present in the target cell. So generally it acts through the GQ protein, and on activations of this one GQS protein, it activates the another enzyme or protein called as a phospholipase C. So on activations of phospholipase C, it cleaves a phospholipids present in the cell membrane, so which produces the 2 secondary messengers. So one is diacyl glycerol and another one is inositol triphosphate. So IP3. So activations of these 2 second messenger, that is the diacyl glycerol and inositol triphosphate, they causes a increasing intracellular calcium level in the cell. So and then this calcium dependent this protein, they got activated. So the main function of oxytocin is, that is the contractions of uterus. So this oxytocin, it stimulates uterus contractions, and the stimulations of uterus contractions by oxytocin is depend upon the presence of the estrogen and progesterone hormone. So if the uterus, it contains more amounts of estrogen, then it increases the contractions of uterus, while the high concentrations of progesterone inhibits it. So during a childbirth, so that is the parturition. So that time what happened? There is a decrease in the progesterone hormone and increase in the estrogen hormone. So due to that, what happened is the oxytocin causes contractions of uterine muscle. So because of this belief, what happened is oxytocin is generally used for the induced labor during a term. So apart from these contractions of a uterus, this oxytocin also helps on the contractions of the myoepithelium of mammary glands and help with milk excretions. And generally, these oxytocin, these secretions of oxytocin is released. It is triggered by a suction of a newborn on the nipple. So which is also called as a neuroendocrine reflex. So this unlike prolactin, the oxytocin response does not decline until 6 months of postpartum. So provided breastfeeding continues. So generally, what we studied in the previous class, that the concentrations of prolactin hormone. So that is that starts fall after the 8 to 10 weeks of the postpartum. So, but this oxytocin, it does not decline. And another thing we have to remember that this prolactin is involved in a milk formation, and this oxytocin do not have any role in a milk formations. And this oxytocin is only involved in a milk ejaculations, so milk excretion. So apart from these uterine contractions and galactobiotic, that's the milk ejaculations, this oxytocin hormone. So it is also involved in the stimulations of follicle luteinizations and steroidogenesis in the ovary. So it is also secreted during a coitus by the female uterus, which promotes the aspiration of semen into the uterus. And during this follicular stage of menstrual cycle, the female, they have a higher concentrations of this estrogen, and estrogen, it promotes the secretions of these oxytocins, so which helps the aspirations of semen into the uterus during a coitus. So in mean, this oxytocin stimulates the spontaneous erections. So clinical aspects of oxytocins, as it mentions earlier, these synthetic analogs of oxytocins. So that this is syntocinon and syntopentane. So they are generally used to induce labor at a term, phenylalanine and arginine. So this vasopressin, it is also called as an antidiuretic hormone, since it prevents the diuresis. And this vasopressin is in human beings, it is also called as a arginine vasopressin, since this vasopressin, it also contains a arginine. So they are also called as a arginine vasopressin. So differentiations between this oxytocin and this vasopressin is, that is the in third positions of oxytocin, that it contains a isoleucine, so which is replaced by a phenylalanine in case of a vasopressin. And in the 8th positions of oxytocin, it contains leucine, which is replaced by arginine in a vasopressin. So because of the presence of this arginine in a vasopressin, this human this vasopressin is also called as a arginine vasopressin, and both have a cyclic structure. So the cyclic structure is formed by the first cysteine and the 6th cysteine. So due to the formations of a disulfur bond between the first cysteine and the 6-cysteine. So like oxytocin, it also circulates in the blood as a free form, and the half-life of vasopressin is around 15 minutes. So there are three types of vasopressin receptors. So B1A receptors, B1B receptors, V2 receptors, and this B1B receptor, it is also called as a V3 receptors. So B1 receptors generally they are generally involved in the this vasoconstriction effect. That means they regulates the blood pressure. So pressure effects, and these V2 receptors, they are generally present in the kidney, and they are involving in the maintenance of this water balance. And B3 receptors, so generally it is present on the anterior pituitary and involved on the releasing of this ACTH hormone. Depending upon the these receptors, where this so which receptor that vasopressin acts, the mechanism of action is different. So generally this B2 receptor, they are associated with a this GS protein, so which stimulates the adenyl cyclase enzymes, and these adenyl cyclase enzymes in turn it increases the concentrations of cyclic AMP. So that is the second messenger, and this cyclic AMP it so it this cyclic AMP it activates the protein kinase, and which phosphorylated the targeted protein, and this B1 and the V3, so they generally act via the they are associated with a GQ protein, so which activates the phospholipase enzymes, and in turn it increases the concentrations of calcium, so it increases the concentrations of intracellular calciums. So B2 receptors in renal tubules, so it stimulates the adenyl cyclase, leading to increased cyclic AMP. B1 receptors and B3 receptors, they are generally present in the non-renal tubules, so which stimulates the GQ protein coupled with a phospholipase C, and this in turn, when this phospholipase C is activated, then it increases the concentrations of the two second messengers, so diacylglycerol and inositol triphosphate, and this diacylglycerol and inositol triphosphate, it increases the concentrations of calcium in the cell. So calcium mediated contractions is contractions of smooth muscles is activated by a B1 receptor. So secretions of this one vasopressin is stimulated by increased extracellular fluid osmolality, so hypovolemia, stress, exercise, drugs like nicotine, carbamazepine, and the secretions of this vasopressin is inhibited by decreased ECF osmolality, so increased ECF volume, and alcohol. So metabolic role of vasopressin. So the major function of this vasopressin is anti-diuretic actions. So generally it causes the reabsorption of water from distal tubules and collecting ducts. So for that, as we discussed in earlier, what happened is vasopressin, it bind onto the specific this one B2 receptor, which is present on the distal tubules and the collecting ducts. And this when this B2 receptor is activated, it activates the this adenyl cyclase, and this adenyl cyclase, it increases the concentrations of cyclic AMP, and this cyclic AMP, it activates the protein kinase A, so which activates the aquaporin 2. So this aquaporin 2 is the water channels, so which is involved in the this transport of water from the this lumen to the cell, so which causes the reduced volume of water in the lumen. So apart from this reabsorption water from these distal tubules and collecting ducts, these vasopressins, it also decreases the glomerular filtrations rate, so which helps reducing diuresis. So along with the other factors such as this renin-angiotensin-aldosterone system, natriuretic peptides, these sympathetic nervous systems, so it collaborate in a maintaining body water system. And generally it is osmolality, so it's depend upon the presence of the sodium, and these regulations of sodium, that is done by the aldosterone mechanisms, and overall the mechanisms of these renin-angiotensin and aldosterone systems, it maintain the body water. So another functions of this vasopressin is it increases the permeability of medullary collecting ducts to urea, lead to the retentions of urea, and subsequent contributes to hypertonicity of the medullary interstitium. So when this in presence of this vasopressin, when this water is reabsorbed from these collecting ducts, medullary collecting ducts, so along with the water, some amounts of this urea is also reabsorbed from the lumen of collecting ducts, so which contributes to the hypertonicity of the medullary interstitium. So because of these reabsorptions of urea from collecting ducts, this urea clearance test is generally it is not reliable as this creatinine clearance test for the test of these glomerular filtrations rate. So pressure effects. So apart from this maintaining of water balance in the body, this vasopressin, they also have a powerful vasopressor actions, so increasing our total peripheral resistance and the blood pressure. And generally this vasopressin, it shows these vasopressor actions by binding to the B1 receptor. So after the binding of ADS to the B1 receptor, it activates the this phospholipase pathway, so which increases the second messenger, this diacylglycerol and inositol triphosphate, and this diacylglycerol and inositol triphosphate, it increases the concentrations of calcium in the cell, and this calcium, the increasing calcium, it made it after the increasing calciums, this calciums it causes the contractions of the muscles, which is calcium dependent. So other actions of vasopressin through B1 receptors are the stimulations of prostaglandin synthesis and the activations of liver glycogenolysis of glycogen. So in addition, this vasopressin can bind to the third type of receptors. So it is B3, or this B3 is also called as a B1B in some books. So then, which is associated with a GQ protein. And generally, these receptors are localized in the pituitary gland. So on pituitary gland, this ADH enhances the actions of corticotropin releasing hormone and contributes to the release of adrenocorticotropin hormone, ACTH. So disorder associated with a vasopressin release. So increased secretions of antidiuretic hormone or vasopressin. So which is this term is called as a syndrome of inappropriate secretions of ADH, SIADH. So in these conditions, the mechanism of hormone secretion regulation is altered. So we know that most of the releasing and secretions of the most of the hormones, so they are controlled by the feedback mechanisms. So in this case, that is in this SIADH, this feedback mechanism is altered. So it cannot be controlled by the feedback mechanism. So that's why there is a continuous secretions of this antidiuretic hormone. And generally it occurs in patients with lung cancer, pulmonary tuberculosis, severe trauma, and other conditions. And because of this excessive, because of the increased concentrations of ADH, it lead to the excessive water reabsorptions and decreased plasma osmolarity. So reduced secretions of vasopressin. So this reduced secretion of vasopressin or antidiuretic hormone, so that causes a decreased reabsorptions of water, so that it causes a diuresis. And the term used for the hyposecretions of ADH is called as it is diabetes insipidus, and this diabetes insipidus is generally characterized by a excessive diuresis. So which means that is a high volume of urine with a low osmolarity. So don't confuse with this one, diabetes insipidus with diabetes mellitus. So in this diabetes means that is generally associated with a polyuria. So mellitus, diabetes mellitus, so that means polyuria with glycosuria. So that means which contains a glucose, and this diabetes insipidus, that is the polyuria with low osmolarity. So tumors like this one, the craniopharyngioma, and other disorder of hypothalamus. So that causes diabetes insipidus can cause hypopituitarism too. So this diabetes insipidus, which is associated with a kidney, is called as a nephrogenic diabetes insipidus. So this nephrogenic diabetes insipidus means that is generally the diabetes insipidus due to the altered in a kidney. So nephrogenic diabetes insipidus. So generally in most of the cases of nephrogenic diabetes insipidus, the concentrations of vasopressin, it may be high or normal in the circulations, but due to the loss of receptors or due to these other causes, this vasopressin, they cannot respond in the kidney. So which causes a diuresis. Though in circulation the level of vasopressin may be normal or increase, but it is unable to show the effect in the kidney, so which lead to the nephrogenic diabetes insipidus. And this nephrogenic diabetes insipidus, it is most commonly seen in chronic renal diseases, particularly those affecting the renal medulla and the collecting ducts. And sometimes in rare cases, this nephrogenic diabetes insipidus, it may be the hereditary. So generally this hereditary nephrogenic diabetes insipidus, it is generally due to the defects in the gene coding for the V2 receptor. So that lack of V2 receptor, or with the mutations in the gene for a aquaporin 2. So this aquaporin 2, so that is the channel which is associated with a water reabsorption. So when there is a mutations occur on a gene that code for a V2 receptor and the aquaporin 2, that lead to the hereditary nephrogenic diabetes insipidus. And in some cases, this nephrogenic diabetes insipidus, it may be the idiopathic. So especially in those patients on which the continuous lithium treatment is going on, so it may cause a nephrogenic diabetes insipidus, or it may be of an unknown cause. So okay, this is all about these hormones secreted by a posterior pituitary gland.